think you've invented the field of mitochondrial transport, right? Like, we invented, so we are the inventor of the field of mitochondrial transport, and it's really a pleasure to introduce him, and I think we're very happy to have him here. So, take it away.
Thank you. Enormous pleasure to be talking to you all, not only in India, but in this beautiful environment. And I did, in fact, invent the axonal transport of mitochondria right after I invented the internet. There's two reasons it makes sense for me to talk. I'm going to use the microphone. Is it too echoey or is it okay? I have a very non-penetrating voice, so we'll hang. Very appropriate for me to talk about, I'll talk about now, for two reasons. One is I think it fits in very well with the theme of this conference, because my laboratory and I have been thinking about mitochondrial transport for more than 20 years, but it's really just in the last few years that we've gotten interested in disease models. So if you think about the title of this conference, we're interested in the basic cell biology of the life cycle of mitochondria in the nervous system. But we've begun to look at it in disease models in Drosophila. And so there were two components to this. One was that I went on a sabbatical about 8 years ago now to the laboratory of Paramount in Boston and learned how to handle flies. And the other component was that my friend and colleague Bill Sack, who's been very generous over the years in schooling my lab, into how to work with them. So what I want to do is talk about some questions we've been asking in a couple of systems to kind of fill in what I saw when I entered this area as a gap in what we know about the cellular and molecular neuropathology of some degenerative disorders, particularly those that involve Parkinson. It occurred to me when I first started thinking about this, there was an enormous amount of information and potential insight about the etiology of these diseases that came out of clinical observations, and even things like taking patient fibrillary, looking at how things work in addiction. There was also a significant amount of insight coming out of molecular studies, finding the genes that were hit in human neurodegenerative diseases, finding them, sequencing them, looking at domain structure, and looking into what protein sticks to what. And so people offered very reasonable hypotheses based on these two very different kinds of studies. But I didn't see a lot of evaluation, especially quantitative evaluation, of cellular phenotype in intact nervous systems. And so that's what we wanted to do, and that's why we moved to this system that was pioneered by Bill Saxon of looking at the intact esophageal nervous system. So, what are we interested in about the life cycle of mitochondria in the nervous system? Obviously, their movement and distribution, so their axonal transport and how neurons get them to the right place at the right time. Their metabolic activity, we can't measure this as directly as we would like. We mostly use dye measurements that reflect the intermembrane potential of mitochondria, although we're beginning to do some things with respirometry, if you will, that are not ready to talk about. And then their proliferation and biogenesis, because remember, this is a very strange organelle, right? It was a respiratory bacteria evolved by a proto-eukaryotic cell, very early in the evolution of eukaryotic cells, and it came to stay. It still got the genome, it still grows, replicates DNA, divides, and fuses as well. And it's very curious how that happens in the nervous system. So we've been interested in these things for a long time, and as I just outlined, we got interested in what aspects of this life cycle are missed in degenerative diseases of the nervous system, many of which are thought to have their focus, or one of their foci, in the mitochondria. So we've had a good introduction to the neuron as an architecturally challenged cell. So I'll just put this slide, accurate representation of the human motor neuron here to remind us you have an enormously long cell. It's got a region with the nucleus and the Golgi and the dendrite specialized for receiving information that can be very different metabolically from the axonal compartment, which can be very different metabolically from the nerve terminus. And then in addition, of course, in mammalian systems, we have nodes and run and along the way. So my contention would be that the need for mitochondrial function, in effect mitochondrial function, is not distributed uniformly over this enormous distance. And so the question of transport and docking and so on becomes relevant. So for a long time, before we began working in flies, we studied the transport of mitochondria in the culture of neurons from chicks and mice, and we came really upon several major conclusions over the years. This is just a chick VR genome. Some of you work on these yourselves, staining the vital dye so that we can see mitochondrial transport. Like in all of the neurons, these neurons have a anterograde moving population of mitochondria and a retrograde moving population. They spend a small amount of their time going in the wrong direction, so to speak, single-digit % of the time. So anterograde and retrograde movement stops and starts in modest changes of direction. And major shifts in mitochondrial distribution can be induced by local physiological activity of growth hormone, signaling along the way, and we now know activity in those around the egg, something we never measured correctly. In the culture of neuron, we found over the years that most of the shifts in mitochondrial transport resulted from the regulation of anterograde movement rather than retrograde movement, and the balance between stationary and motile states. And as you've seen in Bill Saxton's movies from his previous talk, and you'll see in a few minutes, and you can see here in this cultured neuron, in any given time, many, if not most of the mitochondria are persistently stationary. Okay, I'll go through this quickly because you've seen it before. You've even seen Bill Saxton's beautiful diagram before. This is from 2005 review. Mitochondria move in the two directions on microtubules using dynein and kinesin, and they also have myosins, probably in particular myosin 5, which can engage with actin to produce a result that's still not completely resolved. There are also almost certainly docking proteins that anchor mitochondria when these two are in play. All right, so disease models, what's the deal here? The first fly disease model that we looked at was a fly model for Friedreich ataxia. So let me say a couple words about Friedreich ataxia. This is a Diamondback neuropathy, a human neurodegenerative disease with sort of early adult onset. It's an ataxia, so it involves the early in the degenerative process, loss of sensory neurons so that you can't feel your teeth. So you have the problem with gait that's referred to clinically as ataxia. The mutated protein is frataxin, which is something the study has not been really described in very satisfactory biochemical detail. There's a sort of low-resolution structure. What is known is that it's almost certainly an iron chaperone, and it supports the assembly of iron-sulfur complexes for enzymes. There aren't very many enzymes that have iron-sulfur complexes, so we're mainly talking about a protein that supports the assembly and maintenance of elements of the electron transport chain in mitochondria. The known hits in human familial Friedreich ataxia include some point mutations, really quite a few actually, as well as an unstable GAA repeat in the first intron that's very interesting to people who think about plant structure of the genome. The affected cell types are sensory and motor neurons, cerebellum, and then there are cardiac symptoms that I won't discuss. So the cellular phenotype, decreased activity of the enzymes used for structure maintenance, is thought to be supported by frataxin, and the gross phenotype I just described, an ataxia, Diamondback neuropathy, and cardiomyopathy. All right, so the current thinking when we started looking at this about Friedreich ataxia was that a deficiency in this protein through mutation or deletion resulted in abnormal assembly of these iron-sulfur complexes, limiting the effectiveness of electron transport. And in particular, something that has been, I would say, overwhelmingly a dominant hypothesis in this field is not only do defects in electron transport inhibit the normal function of the mitochondria by diminishing its membrane potential, but they can, under some conditions, lead to increased reactive oxygen species production. And this has been what people kind of come from the clinical end have talked about endlessly, that ROS production and oxidative stress are the cause of the neuropathology. And that the pathology of normal death are due to oxidative stress. This idea is actually that the patient's being fed antioxidant compounds I would point out to no avail whatsoever. But other data, including a knockout mouse, had suggested, kind of off in the left field of this area, that there may have been oxidative stress independent mechanisms producing this pathology. Yes? I'm sorry, I can't make you out for that. Does it have any measures been taken? Yes, patients.
Are you saying that the cell taking measures to counteract the ROS?
Does the cell take any measures? Well, no one's really evaluated at the cellular level, so whether or not there's compensation for that is not clear. This is a good point, time to point out tangentially something that Scott Brady used, and that is, from a cell biological point of view, I find it very hard to believe that in a cell, let's say in your brain, that has been alive for 60 years carrying out its function, that suddenly something was wrong. And so one of the background concepts in the work that my lab has done in the last few years is, let's look for what's wrong all the time. And that either must reach a threshold late in life, or as Scott pointed out, you may have a compensatory mechanism that breaks down late in life. So compensatory mechanisms are an interesting thing to think about.
The period to cell composition don't like the superoxides are immediately released in oxygen using dismutase, maybe that's what it is.
Well, it's not even the cells look like it. The half-life of superoxide in aqueous solution is incredibly short before it produces hydrogen peroxide. So yes, there are compensatory mechanisms for dealing with reactive oxygen species. The idea in this field has been that a disorder like this overwhelms them. So what goes wrong in the mitochondrial life cycle when neurons lack the tax? For the study, we used a knockout strain generated in the Anderson lab at the University of Guelph that produces a hairpin RNA that greatly reduces the concentration of the frataxin in these cells. So we carried out this work in the horizontal larval system that Dr. Saxon has described. This is just some experiments we carried out simply to confirm that when we took the frataxin knockdown and we crossed it into the mitochondrial GFP background, that we still have a late larval dynamic neuropathy. So these are just experiments in which we fixed larvae early and late in development and looked at the diameter of the longest segmental nerve. So if we're having a dynamic neuropathy in the distal part of this nerve, the diameter should go down, a very simple measure. And sure enough, we found that while the diameter of this stays constant over time in space in the wild type, that late in larval development declines significantly, indicating a loss of axons. I'm getting there. Yeah, I'll get to transport in a minute.
Do the mutant forms have the same phenotype?
No, we've only looked at this at the now. Yeah. I don't think there are mutant plaques available. We also looked at the cell bodies and the ventral ganglia, which you probably have heard is the magic excuse for a brain and central nervous system. And we have a loss of motor neuron cell bodies as well, right here. Again, late in larval development. So early in larval development, no dynamic neuropathy, no loss of cell bodies. Okay, so now let's get to the interesting experiments. What we wanted to measure in this knockdown stream, where we can see mitochondria, because they're full of GFP, and the motor neurons. We wanted to look at the axonal transport of mitochondria, the resulting distribution and density. We wanted to make this diagram measure of the membrane potential, and also try to get an optical measure of reactive oxygen species levels. And the way we approached this was to look across both time and space. So we looked at 3 different developmental stages: the second instar larva, the early third instar, and the late third instar, so over time. And also, we evaluated most of these parameters in the cell body through 3 different fixed locations on the axon, 3 specific segments, and then the neuronal skin junctions. So across time and across space. You've seen this, I guess, already before I arrived from Dr. Saxon, but what are we talking about? We're talking about making quantitative measurements in live neurons that are still part of the intact nervous system, something that's not that hard to do. those activities you do, it would have been like a dream 10 years ago when we were looking at neurons in the picture. So this is a sort of open larva, so it's alive, but its body wall's been open because they'll let you do anything to a maggot, like there's no animal forms to pull out. This is the ventral ganglion, and here are the segmental nerves running out to innervate the muscles of the body wall. So you can see the segmental organization here, and we work with the longest segmental nerve. So if you zoom in on a particular region, and again, this is something I assume you saw before. So if we zoom in on a single one of these segmental nerves, which represent bundles of tens of axons, and we use the confocal microscope, and we partially bleach a region. We did just a very light bleach to get the stationary bright mitochondrial signal out of the way. We can then follow anterograde transport, which is from left to right. We always organize these with the central nervous system and the cell bodies on the right, or on the left, right? The synapse is out there, so anterograde movement in this direction. We can quantify retrograde movement in this direction. We can look at velocity and duty cycle and flux and so on. And in the same preparation, we can apply voltage-sensitive dyes, ROS-sensitive dyes, and so on to make the measures that we come. Okay, so axonal transport. The first thing we did was simply to get normative data to confirm the work Aaron Pilling and others in this axonal lab has done. And that is to look, there's sort of two ways to observe the properties of transport of these mitochondria. One is to take a particular developmental stage, so this would be late third start L3, and look at three different places. So at a given point in developmental history, how does transport varies apart from the cell bodies, or to take a particular point, this is segment 87, the distal region of these axons, and look at how transport changes as we move through time. So these are just still images from the mitochondrial fluorescence taken again in an intact live nervous system with the confocal microscope. So we look largely in this normative data at two parameters that were not velocity, because velocity doesn't change very much when you develop. One is mitochondrial flux, so this is just a measure of how many mitochondria pass a point per unit time. I like using this measure because if I'm the synapse, I'm the neuromuscular junction, and I'm waiting for mitochondria to arrive, and I need 3 per minute, I don't really care how they got there. I don't care if they went slowly or moved 100% of the time, or if they went quickly or moved 10% of the time. The number arriving per unit time is really important for the future. We also usually, to get more of the mechanism of how changes in flux might occur, we look at the percent of the total time that they're moving for their duty cycle. And so you can see if we look at a given stage in development and look from the proximal to the middle to the distal axon, we see a decrease in mitochondrial flux for both directions of movement. If we look at the percent of the time that they move, again we see a modest decrease as you go farther from the cell body. So normal healthy fly farther, the farther you get from the central nervous system, the less mitochondrial movement there is. But if you look across increasing times in development at a given point, you see a modest increase in both flux and duty cycles. So this is the background, very large amount of data against which we compare the features of the mutant fly. Now, I like to disprove things rather than prove things, and so we had a set of expectations based on people's thinking about this field, prevalent ataxia, and its tendon neuropathology before we went in. What did we expect? Well, it's a diabetic neuropathy. So when you think about mitochondria, we expected some kind of failure to deliver mitochondria to the synapse. That might cause a diabetic neuropathy. So we looked at axonal transport exhaustively across space and across time, and we found that for most of the nervous system, most of the axon, for most of the development in the prevalent ataxia knockdown larva, transport of mitochondria is fine, no problem. Anterograde fine, retrograde fine, duty cycle is normal, flux fine. Until you get to the late stages of development, just prior to the dynamic neuropathy. So later development, out of the distal axons, we did see a difference. And this is just some stills from the movie. You see the bleach spot here, and it's recovery of mitochondria by anterograde and retrograde transport from the wings. And you see the frataxin knockdown flag until there's fewer mitochondria. So we looked at flux, and with flux, there's between the wild type and the frataxin knockdown, there's a decrease in both anterograde and retrograde flux, although the retrograde flux shows a higher percentage knockdown. There's also a modest but significant decrease in duty cycle, again, the retrograde being slightly greater. So if we look at the effect ratio In other words, what is the quantification of motility in the knockdown divided by the control? How much is it affected? We found that for flux, yes, the anterograde flux was reduced to 80%, roughly at 1K, but the retrograde flux was reduced to 50%. This is not what we expected. This is a preferential decline in retrograde transport. It's not a failure to deliver mitochondria in mass to the synapse. It's a failure to retrieve them. And this is one of those things we went back at it a couple times because we still didn't believe it. But once we believed it, we thought this drives a conclusion that we found hard to believe, and that is the frataxin knockdown larva ought to have not synapses devoid of or deficient in mitochondria, but synapses full of mitochondria, excess mitochondria. Could that possibly be?
What did you mean by cause? I mean, whenever one aggregate mitochondria is moving and it's caused for something to start moving.
Right.
So are you taking that in account or aggregate coming in this cause?
I'm sorry, in account of what?
Can you go back?
Yes, certainly.
So in both the graphs down, so you are getting in aggregate and retrograde, there are like causes, right?
Right. This is the anterograde population of mitochondria. This is the retrograde population of mitochondria.
Yeah, so causes in different reasons. So when you say cause, what does it mean?
That's just the percent of the time that they're not moving. Okay, so for instance, in the proximal axon, the anterograde population is spending 80% of their time moving anterograde. They're spending 9% of their time, they're spending 5% of their time moving retrograde, and the difference is the percent of the time they're spending stationary. So sort of the transportology here. The decline in duty cycle here isn't made up by excess time going the wrong direction. It's made up by additional time stationary. So what's happening is you're converting a population that moves a lot to a population that moves a bit less. Does that make sense? Okay. What is what? That's the percent of the time they spend moving. Percent of total tracking time is what we call duty cycle. And you see the three directions here, anterograde, retrograde, and then pausing. All right, so this predicts that we should see excess mitochondria in the synapse. Seemed unlikely to me, but it was an easy thing to look at. We fixed the larvae, and here you see the mitochondria in yellow, and it's the mitochondrial green superimposed on the red HRP-based stain of the neuromuscular junction. So here's neuromuscular junction, the wild type. Here's a neuromuscular junction of the frataxin knockdown. And when we quantified the percent of the area, the fraction of those that were occupied on mitochondria, comparing wild type to frataxin knockdown, early in development, second instar, early third instar, no significant difference. But at the late third instar, what we saw as counterintuitive deficiency in retrieving mitochondria in the distal axon. Sure enough, there's about a 1/3 excess of mitochondria there. All right, so how do you cripple a neuromuscular junction by having more mitochondria? Well, an obvious possibility is that they're not metabolically active. And this is actually the gear that we had turning from the beginning. I found it hard to believe that you could have deficient construction and maintenance of the mitochondrial electron transport chain and have the main problem in ROS production. For precisely the reason that you pointed out, there's ways for cells to compensate. There aren't really good ways for cells to compensate for not being pretty good at making ATP. And so the next thing we looked at was the membrane potential of the mitochondria in the wild type knockdown, using not a beautiful direct method, but sort of the best thing we've got, which is a dye ratio. Yes? Altered dynamics? Well, we're looking in the synapse where they don't move around very much. And those measurements I just showed were made from fixed material. But they do show a very interesting difference that I'll come to in a moment here. So how do we make our measurement as a kind of a standard for metabolic activity? We use lipophilic fluorescent cation, one of the rhodamine compounds, tetramethyl-rhodamine methyl ester. It is lipophilic enough to get across membranes, but it has a positive charge. So it partitions strongly into the cell because of the negative potential of the plasma membrane. and then even more strongly into the matrix of the mitochondria because of the high electronegativity of the inner mitochondria. It's extremely non-toxic, doesn't interfere with respiration. It equilibrates in seconds. If you uncouple these cells, it equilibrates as fast as you can look at it. And so what we've done is to basically treat the dye intensity like we treat the ion concentration and apply the neurostimulant. So using a camera that gives us a linear measure, we take the ratio of the fluorescence intensity of the mitochondria to a region of nearby cytoplasm, and we look at the ratio of mitochondria with the cytoplasmic fluorescence, and that has a logarithmic relationship to mitochondrial membrane potential. So what do we find? Very different from transport. The short message is, everywhere within the nervous system, from as early as we can look at development, the mitochondria and the frataxin larvae had a diminished membrane potential compared to wild type. So here's some data just to sort of show you we did it. You see images here of the wild type, the frataxin knockdown. This is the GFP of the mitochondria. This is the tetramethylester signal. Here's the merge. When we did the ratio calculations, we found that in the wild type, there was actually remarkably uniform mitochondrial membrane potential, both across space and across time, developmental time. In the frataxin knockdown, what we saw was significantly reduced dye ratio, reflecting membrane potential, from the earliest time in development at all places in the nervous system, and in particular, and this will be a recurring theme, the later we got in local development, and the farther out, the more distal we got in the axons, the greater that deficiency in mitochondrial membrane potential became. So this is the axons. We looked at the cell bodies, which I won't show. We saw an even more profound difference in the neuromuscular junctions. So here we're looking again at mitochondrial membrane potential using this wild type and frataxin knockdown. Neuromuscular junctions, it's a little hard to see here, but there's a lot of mitochondrial green here that has been turned yellow by superimposition of vital dimexes. And what that's telling us, and we confirm it quantitatively here, is not only is the membrane potential diminished, some of the mitochondria in these frataxin knockdown larvae are completely depolarized. And I should point out, both in intact nervous systems and in my experience with neurons growing in the dish, it's extremely difficult to uncouple neuronal mitochondria. This is the equivalent of the kind of depolarization we would get if we used two different classes of mitochondrial proteins simultaneously. So to knock this down in these experimentally rather than with a mutation, we would have to apply both an uncoupled and a complex number 3 inhibitor. So really a remarkable decline. And it's true across developmental time here. So from the earliest stage of the good luck second instar, the potential is diminished in the neuromuscular junctions, but really diminished to 50% or less. I
just was struck by this correlation of decreased membrane potential and decrease in retrograde transport, which is the opposite of what people have generally. Some people have said in the past that a decreased membrane potential should increase retrograde transport, right?
Well, if you think that a decreased membrane potential reflects mitochondrial senescence, and if you think that mitochondrial turnover occurs mainly back in the cell body, then yes, it should. And I'll actually come to that in a minute, because in the Parkinson model, we also got counterintuitive results, relative to that idea. Yeah. So it is a counterintuitive result.
Would it be risky with doing phenotype rescue it by destroying the frataxin gain into your DFH, IR?
So overexpress the wild type, haven't done that yet. I can't even begin to hear you. Could you stand up? I'm sorry. Could you stand up, please? I can't hear you. There are more mitochondria in the neuromuscular junction, presumably because of the preferential defect in retrograde transport in the distal axon. Yes, the anterograde flux goes down to 80% of normal, but the retrograde flux goes down to 50% of normal. So what we have is a net accumulation of mitochondria in the neuromuscular junction. What's their morphology? I don't think we can say because they end up superimposed on each other. I know I share with Bill with you that you have to be very conservative when mitochondria are packed together or superimposed on each other to draw conclusions about morphotech. Okay, what about reactive oxygen species? As unsatisfying as it is to try to use as your sole measure of metabolic activity, a dye, it's even less satisfying to do that at ROS levels. But there frankly isn't a really good way other than this. So we use two different fluorescent probes to try to detect reactive oxygen species levels. They're both sensitive to superoxide. They both fluoresce more brightly as superoxide concentrations go up. One is a proprietary dye called Mitochondrial. This actually enters the mitochondrion, and so it's reporting ROS levels in the matrix and adjacent to the inner membrane. The other is a classic dihydroidium, which sits out on the cytoplasm and reports there. And then the positive control, again, using the probe, we know it works. And so what we did was simply to apply anti-mycin A, which is known, sort of biophysically demonstrated, to inhibit complex 3 electron transport in a fashion that greatly increases the dumping of electrons onto oxygen by complex 3. So it's known in a specific way to increase ROS production. So if you put that in and your signal goes up, you know that your probe ought to be reporting ROS. The bottom line is we couldn't find elevated ROS levels in any time or place in these axons. I'll show you some data to prove that we did it. So this is the mitochondrial dye. It locates itself in the mitochondria. This is sort of a complicated bar graph. So what we're showing here is, again, across space, proximal, middle, and distal axons, and across time, second instar, early third, and late third instar. We're evaluating the fluorescence of this dye, and the zeros indicate zero antimycin. So the first bar in each of these pairs reflects the signal we get from the unperturbed nervous system.
So mitochondria which are where those mitochondria are known. Based on the electron transport disconnection, here are some dyes, which can activate the so based on those dyes, can you differentiate whether those mitochondria are functional or not?
Are they functional? What we're measuring is the delta psi, the membrane potential of the inner mitochondrial membrane. That's all we can measure, and that's greatly diminished. We're not measuring calcium uptake. We're not measuring oxygen consumption. We're not measuring ATP production. We're using an indirect measure that's for the best we can do. Does that answer your question?
So my question was, there are some dyes which can differentiate the mitochondria. Whose potential difference around the
No, the dye we're using does exactly that. Are you thinking of JC1? Yeah, this is an aggregating dye that's really quite unsatisfactory for making quantitative measurements. I think the one that we're using is the one that the mitochondrial knowledge has their standard for proof. So if we look at the ROS levels as reported by these dyes, what we see in the wild type is a very uniform over time and space fluorescence, indicating we think a fairly uniform ROS level. The second bar in each of these is the positive control. So if you put in antimycin, known to inhibit complex 3, known to produce additional superoxide, you get a brighter signal. And that's reflected here, being brighter than here, and this being brighter than this. When we looked at the frataxin knockdown, then, what we saw was in the unperturbed nervous system. Oh my goodness. Okay, so let me outline the results without pointing a pointer at data. What we found is in the frataxin knockdown larval nervous system, identical apparent levels of ROS, no increase relative to wild type. We found this in axons, we found it in cell bodies, we found it in synapses. We looked very hard, as it's what we sort of wanted to see, but we could find no evidence with either of the fluorescent probes that are sensitive to ROS that the frataxin knockdown flies were producing those reactive oxygen species, not in the cytoplasm, not in the mitochondria, not in the cell bodies, axons, or synapses, and not at any time in development. The one thing we did see that I'll sort of point out in the power perspective is when we perturb them with antimycin, which remember is the positive control, just to be sure that the fluorescence goes up and the dye is working, we got a more extreme response to antimycin late in development and out in the distal axon. This will come up. Suggesting that the unperturbed reader ataxia block doesn't produce more ROS, but that if it's perturbed, it has the capacity to produce more. just glance at my slides. Okay, so this shows that we have the same level of ROS indicated by the dye and the frataxin not known as the wild type. The only difference is that when you challenge the system with anti-myosin, late in development in the distal axon, a big response in the frataxin knockdown class. And as I just described, same thing in the ROS productions, a fairly uniform level through development of reported ROS from the dye, and the same thing in the frataxin knockdown, no statistical difference. But we see that when we perturb the system with a positive control antibiotic, the frataxin knockdown responds in a bigger way. So here's what I would contend based on looking at cellular phenotype quantitatively. We've got mitochondrial memory potential diminished from the earliest time we can look all the way through development, and from the cell bodies all the way up to the synapses. On some level, if you're coming at this naively thinking about mitochondria, it's amazing that they're crawling around and sort of doing okay, and these neurons don't start dying until late 30s. And so it bears on this question we raised a few minutes ago. This isn't something that suddenly arises just before the dying back around, but there's an underlying problem here that's true throughout the life of these larvae. So we've got these transport deficits that arise late. We've got a counterintuitive aspect of this as far as the direction that's preferentially inhibited. And due to these two together, we have our fundamental counterintuitive result that we don't have pure muscle inventions deficient in mitochondria, but they're filling up with metabolically depressed mitochondria. Can't see a difference in ROS production between the frataxin model and the wild type, although it's possible there's something going on when you perturb the system, so that challenge with the complex 3 inhibitor causes later development and in the distal axons a greater reaction from the frataxin knockdown effect. So I guess we would suggest, whether anyone is moved by this or not, in this field, we would suggest that we ought to be thinking about oxidative damage and the mechanism for this neuropathology involving both motility and membrane potential. So what are the implications here? A lot of neurodegenerative disorders have been pegged all in part to mitochondria, and most of the thinking you see, especially in the clinical literature, is about oxidative damage. I think we should be thinking about them as metabolic disorders, particularly in light of the fruitlessness of treating patients with antioxidant therapy. This could involve ATP deficiency, although I'm a little skeptical about that. Obviously, we haven't done this in the intact fly, but people who work with mitochondria in food, where you can do anything you want, report that as mitochondrial membrane potential goes down, the last thing to suffer is ATP production, that you lose calcium homeostasis, even things like lipid synthesis decline for that. So I'm sort of inclined to think that this might be a signaling difference. This is also in line with something you heard earlier from the thinking of Scott Brady and others in this field, that thinking about these disorders as piles of bricks in the road or tires falling off cars, these are analogies that are easy to use. I think we should be thinking about them in terms of other microscopic analogies like motion detectors and firearms. We really should be thinking about signaling, which can change things quickly in a coordinated fashion. Likewise, with ROS, we tend to think of reactive oxygen species as a terrible thing that has to be kept low by compensatory mechanisms. But in fact, one of the newest frontiers in cell signaling is the recognition that ROS species are involved in signaling. So they also have to be at an optimal level. And if they rise, they might rise nowhere near to the level necessary to be oxidizing proteins and lipids, but to a high enough level to screw up ROS-based signaling. All right. So let me go on to a second model we've been working on. This is work mostly carried out in my lab by Swati Devaray. How many people in this room know Swati? She worked in Sonia's lab. She's been in my lab for about 3 or 4 years now. She's been working on Parkinson's disease models in the same system, the fly larva. And we've been looking at 2 different mutants in pink one and Parkin. I don't have to tell this group that Parkinson's disease in humans and animal models usually involves the loss of dopamine or selectively, and the 2 candidate cellular defects people talk about are once again oxidative stress and mitochondrial dysfunction. Some early onset pedigree have mutations in either PINK1, which is a serine 3 kinase, or Parkin, which is a E3 ubiquitin protein ligase-like protein. Work by Leo Blanc and others in flies and some other systems have associated these with mitochondrial turnover, that is mitophagy. And one hypothesis that's quite prominent and that we set out, remember when principal investigators say we, what really means other people. So that's why we set out the test. The idea that PINK1 associates with damaged mitochondria, recruits the ubiquitin ligase, and targets temporal turnover. So people are thinking of these as defects in mitochondrial turnover that give rise to neurons full of senescent mitochondria and struggle. So let me say specifically what the Blanc lab went through using a genetic approach in flies is to put these two genes into a pathway in which PINK1 is upstream and Parkin is downstream, and establish that mutants show a progressive pathology during development. They focus mainly on muscle cells and found that the pathology involved mitochondria. So the question that was asked when we went into this is, does the cellular neuropathology, to look at the cellular level in an arm, in an intact nervous system, and get out quantitative data, does it support these kind of ideas about what's wrong with the mitochondrial life cycle in PINK1 and Parkin mutants? Just to show a cartoon, it's redistant, I'm using regularly. So the current hypothesis involves the serine 3 kinase PINK1 associating transient with the mitochondrial surface, such that in healthy mitochondria, it's constantly turned over, it doesn't have a long residence time on the surface. That in mitochondria that have malfunctioned or senescent in some way, and this is a necessity, a vague idea still, that PINK1 remains as a long residence time on the surface, that it recruits Parkin, the ubiquitin ligase, and that the action of Parkin somehow marks or targets the mitochondria for turnover, presumably by autophagy, mitophagy. If you have a defect in this pathway, then you expect a defect of mitochondrial turnover that could come in several flavors, a couple of which Dr. Saxton has raised. One is, I think what we would expect in this field, if we screwed up mitochondrial turnover, we would expect defects in retrograde movement of senescent mitochondria back to the cell body for turnover. Another thing is, part of this pathway is thought involved fission and fusion, such that when you have a senescent mitochondria that gets marked by this system, it will no longer fuse with the healthy one, that it remains segregated. So if you screw that up, you ought to have longer mitochondria, because you're at the fission fusion balance. And if you fail to retrieve mitochondria from the axon, the bad one, so to speak, by screwing up this system, you ought to have a higher density of mitochondria. And those are things we could test quantitatively. All right, so what's wrong with the life cycle of these mutants? What are we going to see? So what we look for, we're interested in metabolic effects, we're interested in axonal transport, and interested in fission fusion balance, and we have the three expectations that I just outlined. And once again, we're working in the large nervous system. Yeah.
Are these mutations ones that can put down mutations?
Yes, these are common in humans. The PINK1 that I'll describe the data from in a bit of detail now is a deletion generated by Leo Blanc. Okay, so first of all, after our experience with the Friedreich ataxia model, the first thing we looked at was membrane potential. This is all data from the middle of the long segmental nerve during third star. Again, using the dye ratio, the tetramethyl-glucomethyl dye ratio of mitochondrial to cytoplasmic fluorescence, we found when we looked at the wild type, when we looked at the PINK1 deletion, we saw a large difference of 50% difference in the dye ratio. So again, they're falling around, they sort of look okay, it's a very significant partial depolarization of mitochondria. Okay, so a metabolic defect, I guess I would argue, is expected. If you're accumulating senescent mitochondria, the average metabolic activity should go down. What about mitochondrial movement? Very little difference in velocities, but a significant difference in flux. But not what we expected. We expected retrograde flux to be affected because we expected the failure to bring the bad ones back to the cell body. But in fact, it was the anterograde flux compared wild type to the PINK1 deletion that was very effective. There's a small, modest, and statistically not significant decrease in retrograde flux. So we go to the disease model and we have a decrease in anterograde flux. Now, as you'd expect, what that results in is not an increased density of mitochondria, good or bad in the axon, but a decreased density. So this is a quantitative measure of the density of mitochondria in this region, and we've got a small but significant decrease between the wild type and the P1. Because I'm interested in transport and wanted at least get some ideas that could ultimately be tested about how transport is affected, we looked at this flux difference and broke down the behavior of mitochondria to try to get at what's producing the decrease in flux. Most of the difference in flux could be accounted for by a difference in duty cycle. And this is a new way I'm trying to display these data. So here's the nervous system cell lines over here, synapses out here. And what I'm showing here is the anterograde population, so the net anterograde population that's marching toward the synapse. And we looked at the percent of time that population moves anterograde, their right direction, the percent of the time they move retrograde in the wrong direction, and the percent of the time that they sit still. What we found is that the wild type, they spend 70-odd% of their time moving anterograde, single-digit percent of the time moving retrograde, 27% sitting still. And then what happens in the P1 mutant that has this decrease in anterograde flux is a large drop in the anterograde duty cycle of the anterograde mitochondria. That is shifted to stationary time. So they're still moving anterograde, but they're spending a smaller percent of their time moving anterograde versus sitting still. And so the flux is going down.
What is the main function of extension of these pain?
What are the
Low function and low tension of pain.
Well, it's not perfectly clear. It's a serine 3D kinase with several functions now involved in mitochondria. It's known that it spends a very, very long time on the mitochondrial surface, and data from both genetics and protein interaction studies and some immunofluorescence suggest that it recruits the ligase, the ubiquitin ligase, to the mitochondrial surface, as that cartoon is shown. So for the retrograde population, a much smaller decrease in their duty cycle in their correct direction, so to speak, that falls then into stationary time. And this explains almost all of the difference in flux. What about fission-fusion balance? It's really hard to measure. Dr. Saxon and I talk about this a lot, and I'm looking forward to talking to you later. So I just don't know how you're doing it. So what we did is just to measure the lights. And this is for students to get for any of you students who are going to ring the net of cells and get them to pop up quantitative data, or as we say in our we take the cell by the angles and we shake it upside down until the change falls out of its pockets and we get quantitative data. Don't just look at means and medians, because here we have in green the distribution of mitochondrial life, frequency distribution for the pink line deletion, and in white we have frequency distribution of mitochondrial life for the wild type, and the means and medians are pretty darn close. But in fact, out in the wings, there's a population of very long mitochondria, more than twice as long as the population in the P1 mutants. And because this 10% of the mitochondria are a whole lot longer, they represent about 20% of the mitochondrial mass. So this is consonant with the idea that you are perhaps affecting the fission-fusion balance in such a way as to prevent the segregation of bad components. And so in a nutshell, what we've seen so far, what Swati has seen so far, is we've got something we didn't expect, decreased anterograde flux, decreased delivery of mitochondria to the axon and lower density. We've got something we did expect based on the thinking about the cellular neuropathology of Parkinson's, which is reduced membrane potential. And then we have this subpopulation of very long mitochondria that are stationary. What we'd like to pursue directly then is what the fission-fusion balance is, because of course you've got mitochondria that fuse to make bigger mitochondria, and then go fission to make smaller mitochondria. And you'd like to know what's effective to give you a net shift in mitochondrial length that can report either up to results. And then what's the rate and location of turnover? So one possibility is that there's mitophagy out in the axon, and that we shouldn't have expected that. We shouldn't expect them to move retrogradely toward the cell body to be turned over. I am the person who 21 years ago showed that autophagic decals can form out in axons, and yet I find it hard to believe that there's a ton of mitophagy out there. It's just such a big organelle and such a big process. But we'll see. I find it, this is just a gut level thing, so you can ignore it. The idea that there's a ton of mitopathy way out of this axon, I'm not convinced. But that would explain why, when we knock down a system that's supposed to be involved in mitochondrial turnover, we don't see a failure to retrieve them. Maybe they aren't treated sub-optimal. We're actually looking right now in the same system, the plaque level system, at the distribution in live larvae of proteins associated with autophagy, and we actually don't see that localizing mitochondria. So that's right. Yes. How do you detect it? Good question. The simplest way we can think about is to co-express in different fluorescence channels a key autophagy marker like ATGA, which is the one we're working with, and mitochondrial GFP, to look for them to co-localize, to look for mitochondria that are newly sequestered into autophagy factors. That's what we're looking for right now, so far in the scene. Mitophagy-specific markers. I mean, I don't know of any proteins thought to be exclusively involved in autophagy of mitochondria as opposed to more general autophagy. Although those may emerge as people begin to focus on mitophagy specifically. Good point. Not here. I'll show some data on Wednesday that we did in cultured vertebrate neurons that tells us a little bit about the distribution of long mitochondria when you stimulate fusion or inhibit fission. But they're distributed through the genome. Now, this is all a measure of data showing the stationary mitochondria. The moving ones in these neurons are small and it's very difficult to get an accurate length measurement, and so we didn't attempt that. Let me give credit where credit's due, and then I could take some more questions. Like I said, when PI say we mean other people. These are the other people. But the data I talked about primarily was carried out by Yujiro Shidara, who was a wonderful postdoc on my lab, tragically killed in an auto accident a few years ago. We have two terrific technicians in the lab, one of whom is Garland Kutz, has been involved in this project. There's Swati. She look familiar? Does she look happy in America too? She seems happy today, even though it's hard to get in some for breakfast in West Indiana. Swati, and this is Jim Sung, who's working on the Parkinson mutants data that I didn't include in today's talk. So thanks for your attention. Do you have any questions? Yeah, Bill.
So you spent a lot of time looking at TMRM and looking at polarization like the 2 middle axon, the pink one, and things. There's a long-standing opinion out there that low membrane potential drives vector transport or signals vector potential, or vice versa, high membrane potential. But you haven't said anything about correlation between membrane potential and transport direction. Right. Therefore, I know that you saw that. And I want to know from you. I know you're very polite and temporary in your interpretation. But can we just put this to rest?
Yeah, well,
there is no correlation.
Yeah, I'll show data from another study on Wednesday. Now, my reps are really hard because it's such an attractive idea. Think about it. Dream with me for a moment. If mitochondria mostly arise through biogenesis in the cell body, and there's sort of visitors in the axon, and you expect sort of fat, sexy mitochondria outbound with a high memory potential, churning out ATP, they somehow become senescent because different fusions are good enough to redistribute fat stuff, and then the ones coming back with a low memory potential, be metabolic and different metabolic states. It's a very attractive idea. There's evidence out there for using what I consider crafty diets. Thank you, Bill. When you look at it as carefully as you can, we see no correlation between the direction of movement and membrane potential. We see no correlation between being stationary versus moving in membrane potential. We've absolutely looked at this to death. The only things we've seen, and again, I'll show you about this on Wednesday, the only things we've seen in a neuron that modulate membrane potential, the only things, very important things, are cell signals. So in the region of growth, in an actively growing axon, you see a higher impairment potential. In regions of experimentally induced focal cell signaling, you see a higher impairment potential. And so presumably, if someone in one of these wonderful myelinating systems looks at this, maybe in the parajuxtanodal region, might kind of accumulate, nodes of Bombay and active iron, maybe they'll see a difference. But no difference relative to movement versus non-movement or attributed versus movement. We look really, really hard and just can't find it using techniques that we trust.
As you probably know, we did use the MPTP metabolite of MPP plus and see a very striking effect on transport, regenerative transport in general. And although we didn't highlight it, the mitochondria were depleted from those presynaptic terminals as well. Have you looked at that?
No, we haven't used that behavior.
So it might be interesting as a control for activating regenerative transport, because we certainly see, I don't know for sure what will happen.
Oh, I see. So for us, it's interesting. Very interesting. And it should be easy to do.
Right, and in that case as well, one of the things that was interesting about it probably wasn't as, Peter probably knows, but I don't know if everyone does. It is thought that MPP plus is a mitochondrial poison, and that it will create toxicity. Of course, it turns out the oxygen. Aside from that, one thing it does do is that it causes an increase in caspase 3, which is associated with mitochondria as well. And that caspase 3 in this case cleaves a PKC isoform, PKC delta. And it's that kinase which is what's responsible for the activation of regenerative transport. So, I'd be interested in finding out whether or not it does work the same way in Drosophila, or maybe it's a different system. I don't still know about the PKCs there, but yes.
I'll think about that and get in touch with you when we get back. That's an experiment that's worth doing. It shouldn't be that hard to do. Other questions? Well, I guess I'm the only thing standing between you and your lunch.
No, that's not.
Oh, you said.
You all yeah. Okay, so we want to have a good photo because every time we enter together, not really because we have put them in so please assemble outside. Yeah, sure. So just so you know, there is a slight change